Hello, this is Marilyn Ritchie. I'm a professor of genetics at the University of Pennsylvania. I'm here today to introduce you to the Penn Medicine BioBank, also referred to as PMBB, to tell you how you can gain access to these data and describe some of the ways that these data can be used. First, an introduction about precision medicine research. As you may be aware, linking genetic data with clinical data provides great opportunities in precision medicine. One of the ways that has become very common to do this is through the use of medical biobanks. A medical biobank is where we have electronic health records that captures information about patients during their routine clinical care, and a biospecimen repository that includes blood, serum, plasma, and DNA, among other biospecimens. This type of biobank gives enormous potential to do discovery science and also implementation science, where you might try to take research findings and implement them back into clinical care. The data included in a medical biobank can be very vast. We can have biospecimens that can be used for both molecular and cellular assays, such as genotyping, sequencing, transcriptomics, proteomics, metabolomics, etc., and electronic health records, which includes all of the information captured during clinical care. This includes diagnosis codes, so what diseases do people have, procedure codes, which is an indication of what procedures have been done to the patient, medications that are prescribed, laboratory measurements, imaging, and clinician notes. The utility of these medical biobanks is enormous. With all of these data that I just described, diagnoses, imaging, clinical lab measures, these would be things like your metabolic panel, blood counts, glucose levels, and genomic data or other omics data that can be generated, can be used to generate all sorts of discovery science, such as genome-wide association studies and phenome-wide association studies. Here in this top figure, I'm showing an example of what's called a Manhattan plot, where you have genomic locations across the x-axis and a negative log 10 of the p-value on the y. Here, higher means more statistically significant. And then all of these points indicate a particular genetic variant that's associated with a particular disease or phenotype. In the bottom plot, what I'm showing you is a distribution of polygenic risk scores. This is where we can take an aggregate combination of genetic variants across the genome, combine them in a mathematical formula to predict someone's polygenic or many genetic variants combined risk of disease. Here at Penn, we have created the Penn Medicine Biobank, or PMBB. This is a single institutional research program which recruits patients from any University of Pennsylvania health system, clinic, or hospital. So far, recruitment has been from a variety of outpatient sites in the course of their routine clinical care, which means that this biobank is disease agnostic. We're not recruiting specific patients with particular diseases. We're recruiting any patient that's interested. The blood is collected and processed centrally. We're storing DNA, plasma, and serum on all patients. And then we do have select tissues on some patients, such as CSF or actual tissues from surgical procedures. Patients consent to giving access to their clinical data via the electronic health record, and we gain permission for recontact and callback studies through the consent process. As of 2021, we've recruited over 65,000 patient participants into the biobank with biospecimens stored, and we have ongoing recruitment efforts with a goal of recruiting over 500,000 Penn Medicine patients. As I mentioned, the recruitment is broad and disease agnostic, which means that no matter what disease you're interested in, there are probably a number of patients recruited already in the biobank that have that disorder. Thus far, we have 20,000 patients with genome-wide SNP data or single nucleotide polymorphism data, and 45,000 participants with whole exome sequence data. What I'm showing in this figure on the right is an an image of the projection of principal components of ancestry, which gives us an indication of the ancestral diversity of our patient participants. What you can see is that we have a large number of patient participants that cluster here, which is an indication of European ancestry, and then a large number of patients that are along this axis of African ancestry, and also patient participants along this axis, which would be of Asian ancestry. So what I want to talk about now is how can you gain access to these data? As a Penn employee, student, faculty, staff, postdoc, you are able to access these data. First, you'll want to go to the PMBB Data Access website, which can be shown at this URL. To gain access to this site, you will need to enter your Penn key and password. Once you're here, you can click on this project information link, which will then drop down this menu. You're going to want to start with the application form. From there, this data access agreement form is going to appear on the screen. This agreement sets out the rules of the road or the rules of engagement for working with these data. You can scroll through this document and read and then sign it electronically. Once you sign, you'll be given the opportunity to add the names and email addresses for all of the members of your lab or team. This is important because anyone who will access these data will need to sign this form. This is a way to make sure that everyone that's going to use the data is fully aware of the policies and procedures and limitations with using these data. For example, the data are de-identified, which means that we do not know who the participants are. One of the policies is that we cannot try to re-identify the participants. So once you enter the names and email addresses of your team, this form will be emailed to all of them automatically. This form is something that we're doing electronically because we plan to send it out annually and have every investigator who works with the data review and sign each year. Once you've signed, you will be taken automatically to the application form for access. Here, you're going to describe the project, indicate the team members and their contact information, any other investigators at the University of Pennsylvania who will access the data, and then a brief scientific rationale of the project. This is then reviewed by our internal Penn Medicine Biobank review team, and this is really just to make sure that we have the IRB approvals in place and the computational infrastructure to use the data. We're not doing a scientific review and judging who can or cannot use the data. Anyone at Penn has the full right to use the data. Approved projects are given access to genotype and phenotype data. I should specify here that what I'm talking about is the data access process. You can use the same form for specimen access, and that goes through a little bit more interaction with the biobank team to make sure that you can access the specific specimens at the right concentrations and volumes that you need. Once you gain access to the genetic and phenotype data, what you will get are the 20,000 individuals with genome-wide SNP data, and this has been imputed to the Top Med Reference Panel, which is an ancestrally diverse reference data set. 45,000 individuals with whole exome sequence data, and phenotype files that include diagnosis codes, procedure codes, vitals, laboratory measurements, medications, and smoking history. We're currently making these files accessible in read-only directories on the HPC or LPC, which are two of the high-performance compute clusters that are managed by Penn DART, formerly known as PMAX. For other more complex phenotypes, you can reach out to the IBI Clinical Informatics Core or the DAC, and they can help you get access to those data out of the EHR. Now, if getting started, you don't necessarily want access to all of the individual level de-identified data, but instead you just want to interface with the data a bit, we do also have the PMBB Genome Browser. At this URL at the bottom, you can go here and search for your favorite gene or variant, and you can search for the variant by chromosome and position or RSID, which is one of the most common identifiers for a single nucleotide polymorphism. This site does have public access. So this is a way that we are able to share information about the Penn Medicine Biobank with the broader community. And this is important for scientific publication and data sharing. Once you enter a gene or variant of interest, you will be taken to a set of tables where you can look at a particular gene. So here in this example, I'm looking at ApoE. Each position or RSID with the chromosome and base pair location, the reference allele, the alternate allele, some annotations about that variant, a Revel score, which is an indication of functional impact of the variant, and then allele frequency. So we have the overall frequency as well as the ancestry-specific allele frequencies. This is a great way to get started with the data if you've never worked with genetic data before, but you want to get a sense for what we have captured in the biobank. I'll also mention you can look at either the whole exome sequencing data or the imputed data, depending on whether you're most interested in coding variants, which would be the whole exome sequence data, or non-coding variants, which would be the imputed SNP data from the GWAS panel. There are a variety of ways that these data could be used. As I already mentioned, genome-wide and phenome-wide association studies, where you can look for the association of specific variants with traits. You can do polygenic risk scores and even a polygenic risk score PheWAS, where you calculate the polygenic risk score for one trait, say, for example, hypertension, and then you test for the association of the hypertension polygenic risk score with many other traits across the EHR. You can also do analyses of imaging data. So this example here is showing an example of quantitative measures of hepatic fat and then testing for the association of hepatic fat with traits across the EHR. You can also look at rare variant association. So here, this is doing a gene burden test, which is where you combine loss of function variants across a gene, and then you test each gene for association with a phenotype. These are just a small number of examples of the types of things that you can do with these data. In the future, we will be launching a new PMBB website, and this will have sections for the patient participants as well as for researchers. We're also going to be launching a PheWAS browser, where we have already conducted a common variant as well as a rare variant gene burden PheWAS across the Penn Medicine Biobank participants. And soon you'll be able to search and look for those associations. So again, if you're a researcher who really doesn't want to learn how to run these analyses yourself, but instead just wants to be able to look at your favorite disease or your favorite gene and find out what associations there are in the biobank with that gene or with that disease, we're going to be able to search that very soon. And as I mentioned earlier, we are continuing to expand the PMBB. We want to recruit more broadly across Penn Medicine, including many of our outlying hospitals. We want to generate more genetic data, both in the form of the GWAS SNP arrays as well as whole exome sequence. And we're starting to engage more with the participants to collect more data. This would be things like surveys, wearables, and thinking about geocoding to link the locations of where participants live with public databases regarding social determinants of health. Finally, I want to acknowledge all of the people that make something like this possible. This takes a village to get something like this pulled together for the community. First, the Penn Medicine Biobank team, the Penn Turbo team, who are doing some of the phenotype definitions, many of the departments and institutes and centers across Penn Medicine. And finally, if you are interested but have more questions, feel free to reach out by email to myself, Marilyn Ritchie, Dan Rader, or Joellen Weaver, and we will be happy to help you. Thanks so much for listening, and I hope you enjoy working with the Penn Medicine Biobank.